Hello. Today we're going to go through the Engineer It Kit experience using the 4-day schedule, which is by far the most used for Engineer It kits, especially in the classroom, because it fits nicely into the 60-minute periods. Today we're going to be using the individually packed Engineer It kit, but if you have a group kit, it works very similar, and I'll describe a little bit more about how the group kit works as we go. And we're also going to use the DNA Playground by Amino Labs. This is the classroom edition, and that basically means it has a little more incubator space than the home edition. Alright, so generally how it works is day one we're going to be making Petri dishes with LB agar. That's the stuff that the bacteria grow on and eat. So that they can grow and divide. And then we're going to streak some bacteria onto those plates so that they grow and divide for tomorrow. On day two, we will collect some of those cells and we will actually go through the process of engineering them. And we'll get to a step that we call the recovery step, where you, after you've gone and engineered them, you need to bring them back to life and get them growing and loving life again. And then on day three, we're going to actually take those recovered cells and we're going to put them onto other Petri dishes that we're also going to make today. And then on day four, we'll actually see the results. Hopefully we'll see some nice colorful colonies of bacteria that are producing a colorful pigment that typically comes from ocean creatures. All right, so let's get going. So these are well sealed. Sometimes they're a little hard to open. I'll pull out all the components here. Okay, I'm going to keep the bag because we're going to put some stuff back in there. So we'll start with this. This is called an Inactivation Bag. So this is a bag that you're going to put all of your used tubes and loops in when you're done with them. You don't need to necessarily put your gloves in here unless you're pretty sure you got bacteria on them. Otherwise, these can typically go into the regular garbage. And so I have a yogurt container here. Just a normal yogurt container. If you have something even a little bit bigger or heavier duty, you can use that as well, but this is a very cost-effective way to prop up the bag. And so you kind of just put the bag in to the yogurt container, and it'll kind of just sit there, okay? And so you can put your stuff in there as we go through the experiments. Okay, so these are loops, inoculation loops. We've got a couple of different sizes. We've got some 10 microliter yellow loops. Okay, you can see they've got a little loop at the end. That loop can hold 10 microliters of liquid. And we also have these blue loops, which are much smaller. They can hold about 1 microliter. Okay, today we're going to be using some of the yellow loops, but in the future days we're going to be using some of the blue ones. So in fact, today we're going to be using 3 yellow loops. I'm going to put the rest back. Actually, we're going to be using 4 today. Okay, I'm going to put the rest back. Next, we have Petri dishes. Again, this is what we're going to be putting LB agar into, and this is where the bacteria are going to grow. Okay. We've got sterile water, and this is what we're going to mix some powder into to make that jello-like LB agar. We've got a stencil, which we're going to use today to help us streak cells so that we can get some colonies. And colonies are these little mounds of cells that are growing, and the goal is to get some of these mounds of bacteria or colonies so that we can engineer them tomorrow. Okay, we have the day one bag, which we're going to use today. This comes with some blank cells. These are cells that haven't been engineered yet. You can learn about them in Chapter 3 of Zero to Genetic Engineering Hero. We've got some LB agar powder, and again, this is kind of like Jello. We're going to mix it in the water and then pour it into these plates. And then we've got an antibiotic pill here, and that's because when we do genetic engineering, often we use this methodology called selection so that we can select for our engineered cells. And I'll talk more about that as we go through. This is the day 2 bag, so we're not going to use this today. We're going to be using it tomorrow on day 2. In general, it comes with some positive control cells, meaning cells that are already engineered, so you can compare them to your cells that you're trying to engineer. We have a buffer called T-buffer. This is used to help put DNA into the bacteria, right? The process of genetic engineering. We've got some DNA, and we have some recovery media here, which is used to help bring the cells back, get them growing again after you do the engineering, okay? So we're not going to use this. This is obviously for day 2, but the rest of this stuff is for today. Okay, so the first step is to make the plates, and for that we're going to need the Petri dishes. We're going to label them. We're going to label them with a Sharpie. All right, and when you label plates, you always want to label them on the bottom, okay? So it's important to know that with the Petri dishes, the bigger part, which is going to be this here, is the lid, and then the part that has the star on it, so you can see there's a bit of a star there, that is the bottom, okay? And so ultimately we're going to be pouring into the bottom, okay? And we always label the bottom of the plates because the lids can fall off, and if they fall off, they might get swapped around. And so if you label the bottom, which is where we're going to pour the agar, your label will always stay with the agar, okay? So that's important. So for the engineering kit experience, we have a few different plates here. We're going to have one plate where I'm going to write NS, which stands for non-selective. This means that there's going to be agar in here, but it's not going to have any antibiotic, okay? And this is because we need to grow our blank cells, which aren't engineered yet and don't have any sort of antibiotic resistance. All right, so this plate we're going to be using to streak cells today. Another plate, I'm going to put an S for selective, and I'm going to put a plus next to it. Now, selective again means you've added antibiotics so that you can select for your engineered cells. The plus stands for the positive control, which I mentioned is in the day 2 bag, and we'll talk more about that tomorrow. I'm going to write another S with a minus. Okay, so S minus, this is the negative control. All right, and a negative control essentially is when you do an experiment and you usually remove something from that experiment, and if that experiment runs well, you'll get a negative result. So for example, like nothing will happen, right? And I'll talk a little bit more about that. We're going to do this one today. And lastly, we've got the S, and we're going to put an E next to that for experimental. All right, and this is the selective plate, which we're putting our experimental sample in. So this is going to be our engineered cells. Hopefully we'll see some nice colorful colonies on this in a couple of days, okay? All right, so we've labeled our plates. That's pretty important. So they're ready. And now we can go ahead with creating the LB agar to pour into those, okay? So for that, so we don't need the cells right now. We'll need those in a little while. We can even leave the antibiotic here. But we are going to need this powder, and we are going to need this water. I'm just going to put that to the side, and so we're going to go to the microwave. And before we do that, I just want to stress how important it is to get the water boiling. Okay, so the objective here is to get a rolling boil in the water. Okay, and this is because LB agar is comprised of LB, which is the amino acids, the sugars, the minerals that the bacteria grow upon. So they eat that and they grow with it. And then the agar is, it's sort of like gelatin, except it comes from ocean plants. And in order to dissolve that agar, you need to have at least 85 degrees Celsius water, okay? And so that's really hot. And also when you add the powder to the water when it's boiling, that cools it down. So that's why you need to have hot water, okay? So can't stress that enough. You can't just pop it in the microwave and take it out in 45 seconds, pour it in and expect it to work. You definitely need a rolling boil, and we'll see that momentarily. So let's go do that right now. So I've got a regular microwave. It really depends. The timing depends on the power of your microwave. This is like a regular run-of-the-mill 1,000-watt microwave. And it typically takes 45 seconds to do a single bottle of water. If you have a larger group and you want to do multiple bottles of water, you'll have to add more time for them to work. But actually, it ends up working out to be the fastest in order if you have your students do one bottle at a time. Just form a line, 45 seconds, done is usually the case. Okay, so I'm going to loosen the lid here so that when it boils, the vapor can escape and it won't explode. I'm going to put it in here, and I'm going to go 45 seconds. Try and get close here. Okay, so you can see that it is not rolling. There's no rolling boil yet. So we're going to go another 5 seconds. So there's this very slight rolling boil in this one. We're going to go a little bit longer. You definitely want to see it boiling. All right, so you can definitely see there was a rolling boil there. Let me do it one more 4 seconds just so you can make sure you can see a rolling boil. All right, so you can see the rolling boil there. Okay, so that's where you want it to be. Careful, it's going to be hot. If you hold it by the lid, it's going to be much less hot. We're going to get the agar powder, remove the lid, so there is going to be vapor coming out. We're going to take the lid off, and you kind of need to pour a little bit fast and give it some taps, and you will get some agar stuck to the edges around the rim here, and that's simply because the vapor when you pour it in. I'm just going to put that on. I'm going to put the lid slightly back on, just a little bit, and again it's hot, so you want to swirl it to mix in. You can see there's a little powder on the side there. You don't want to be too vigorous because then you introduce bubbles, and that can disrupt your plates a little bit if you've got a lot of bubbles in there. So this one actually looks pretty dissolved. It's pretty see-through already, but it's a good idea, and it's recommended that you just put it back in for another 5 seconds. And when it boils this time, it's not going to have a rolling boil from the bottom. It's actually going to foam up a little bit. So I'm going to do that. 5 seconds. Okay, there was no foaming. I'm going to do another 4 seconds. There we go. You see that foaming? Now it's going down. Okay, so now we're perfect. This is going to work out perfectly. So now we're going to go back to here. When you discard your tubes, you just take the lid off and put it into your baggie, okay? All right, so we have 4 plates here. 3 of them are selective, meaning they will have antibiotics, and 1 is non-selective. meaning there's no antibiotics. And you might realize we haven't added any antibiotics yet, so we can go ahead and pour the non-selective plate. And if you look into it, you'll see that it's quite clear. That's a good sign. If it's cloudy and you see little like white powder kind of floating around, that means you need to microwave it a little bit more. Because if you just go ahead and pour your plate, if you see that powdery cloudiness, it's not going to turn into a gel afterwards because the agar didn't fully dissolve. Okay, so again, it's a little bit hot, so just be careful. I'm going to pour this so that it's halfway, pretty much up to the star. Okay, and I'm just going to set the lid partially over, and this will help it cool faster, and it'll help for a little bit of evaporation so it doesn't get super humid inside the plate. Okay, so now we can pour in the antibiotic. Okay, so I'm going to pop that lid off and just pour it in. And I'll put this into the baggie so you can see it in there. I'm going to put the lid back on just to help limit contamination. And if it's too hot, you can hold the lid so while you gently swirl. Okay, just need to gently swirl and let it sit for a moment. Gently swirl the other way. Gently swirl. It'll take a couple of minutes for this to dissolve, and the important thing is that the internal powder within this capsule dissolves. Okay, so you might still see some gelatin capsule floating around, but if all the inside is dissolved, you're good to go to pour the rest of these plates. See how I'm not introducing too many bubbles in there? I can see a few. Let's just open it up, have a look. So you can see the pill is starting to dissolve, but it's still a little ways to go. It's almost dissolved. And the cooling period and solidifying period really depends on your environment. If it's very humid and warm, it can take up to half an hour. If it's cool and dry, like it often gets in Canada, it can be only a couple of minutes before they're ready to go. So just keep that in mind. So you can see this is pretty much, I don't see any more components. I see a little bit of gelatin capsule floating around. I'm going to go ahead and pour these now. And so same thing goes. You want it about half full. Okay. That gelatin will dissolve through time, okay? Alrighty, there we go. So I'm going to put these back on most of the way and let these sit for about 15 minutes and they will solidify and we can move on to the next step. Alright, so we're back. It's been about 15-20 minutes. If we have a look at these, they become a little bit, like, not as clear as before, but they're very clear. So these plates are well done. If you flip them, nothing's going to fall out. One thing you can do as you're kind of waiting for them to dry is if you take off the lid and you have it on the table and you give a little tap on the side, if you see it jiggling at all, then you need to wait a little bit longer. Okay, but once they're cooled down to room temperature, they should definitely be solid. All right. Okay, so for today, on day one, we're going to need the non-selective plate to streak blank cells And we're also going to do the negative control plate. And so these other two Petri dishes here, they can actually go back into your Petri dish bag, and they can go into a fridge. And so these are sterile, or they should be pretty sterile. So there's no bacteria on them yet or anything, so generally they can go back into a fridge. Okay, so now we get on to working with cells here. And so the first one we'll go ahead and do the non-selective plate first, where we streak blank cells, okay? And so again, these are E. coli cells, lab E. coli cells. Again, you can learn about them more in Chapter 3 of the Zero to Hero book. But effectively, they're a basic lab strain that is not engineered with any sort of plasmid. And tomorrow we'll be engineering with plasmids, and we'll talk more about what they are, but basically they're just a little loop of DNA, a very small loop of DNA that in general has a gene that you want to express, such as a colorful pigment. It'll have a gene for some sort of selection, okay, so that the organism can become resistant to an antibiotic. And then they have a little segment called an ORI, or an origin of replication, which tells the cells to copy them, okay? So the little plasmids are what we're going to be putting into these cells tomorrow once we have colonies, okay? So in order to get colonies of bacteria, we'll do what's called streaking, okay? And essentially the idea is there's a lot of cells in here, and they're all kind of located mostly at the top, so you don't need to like dip or like scoop anything out. As you'll see, you just need to dip a loop in. But the loop will be covered in lots of cells. And what happens is if you put lots of cells onto a Petri dish, you just get this lawn, which is basically like a thick layer of cells, all right? But we want colonies. We want these little mounds of cells because they take in DNA better, all right? And so in order to get these colonies rather than the mounds, we're going to do a streak method where we're going to dip a loop in to the cells, and then we're going to spread it onto the Petri dish on this line 1, okay? And then we're going to discard that loop, and we're going to get a new clean loop. And with that clean loop, not dipping it into the cells the second or third time, we're just going to rub that first loop through the first line and do another zigzag. We're going to throw that loop out, get another loop, and do the same thing. Drag it through the second line and then do a few zigzags. Okay, and the idea is that because on the second line here, you're only dragging through this small part of the first line right here, you're only collecting a small number of cells and then you're spreading them out through a larger distance. And then even more so on the third, you're dragging through the second line, so even fewer cells, and then streaking them out, okay? So you get less and less cells spread out over the Petri dish, and then as they grow, that single bacterium will grow and divide into thousands and thousands that will form these little mounds we call colonies, okay? And then we'll be collecting those on day 2 for engineering, okay? So let's go ahead. You can put your non-selective plate on top here. Because I'm left-handed, I usually put the line I need to draw on the right-hand side. And you can open up these by simply kind of pushing so that the back part pops out, and then you can pull it out. This is a loop, it's sterile. You don't want to touch this end, you don't want to touch it to the table, you don't want to touch it to your hands or your body. Okay, so keep that sterile. Open this tube, and again, you're just going to dip it in gently a few times. You don't go too deep, you don't need to scoop. If you look at it and it's wet, which mine is wet, then you know you've touched the bacteria inside and you're ready to streak, okay? So I'm just going to, at a kind of a strong angle here, kind of like maybe you'd paint something. I'm just going to rub across this first line. You can just go A to B if you want, or you can just go back and forth a few times on that line. Okay, make sure you get lots of bacteria on that first line. That's fine. Okay, and then I'm going to put that into my discard container. I'm going to get a second loop. I'm just going to turn this a little bit, and you can either follow the line exactly, or for me, I'll drag through and I'll do a few more lines like this. Okay, so now I just dragged a little bit of bacteria in that line and spread it out over this whole area here. Now I'm going to put that in my waste container. Now I'm going to get another one, clean loop, and again, I'm not dipping the loop in after the for the second and third time, okay? Because if I did, then I would have lots of cells on here again, and it would defeat the purpose of trying to get less and less, right? So same thing here, I'm just going to drag through and do a little zigzag there, okay? Done. Put my lid on, put this back in the inactivation bag. I'm done with, oh no, I'm not done with these cells yet. I got one more thing to do. Okay, so now we have the negative control, okay? And the idea here, so controls are often used to try and understand your experiment, make sure things are working properly in your experiment. And so in this case, this is a negative, we're going to do a negative control where we try and gauge whether the antibiotics were properly added to this Petri dish, okay? And we call it a negative control because we're going to be streaking some blank cells which aren't engineered, okay? And so they lack a plasmid, they lack the antibiotic resistance that should, and so they should be susceptible to the antibiotics in this agar. So in other words, if the experiment works well, there should be no growth on this plate because these bacteria won't be able to survive, all right? If, let's say, you forget to add the antibiotics, or let's say the antibiotics are bad, or you know, something just went wrong where the antibiotics aren't in here, or they're in here in a low level, you could actually see cells growing on here, and that would tell you that something is wrong with the selection in this plate because blank cells should not grow if there's the right amount of antibiotics, okay? So that's why it's a negative control, because we expect things not to grow. So I'm going to take a single loop for this. We're not trying to get colonies or anything. We're simply trying to see whether the cells will grow on this or not. So you're going to, again, dip it in here, make sure it's wet. I even got a little bit of agar on it that time, which is fine, but it doesn't matter. And I'm just going to drag this all the way across like that. Make sure you get cells on there, okay? There should be no question as to whether the antibiotics work or not. So let's put lots of cells. All right, and then that goes in the container. All right, so now I have my two dishes that have cells on them. I'm going to take my stab of cells. I'm going to put that in the waste container as well with the lid open. You put the lid open so that later on when you add bleach, the bleach will be able to get into the tubes. Okay, so now we're at the DNA playground here. Just really quick about the playground. You want to put it on a flat surface. On the screen here, we've got a timer. If you press go, and you move over, it'll keep going. We have two stations on top, a cold station and a hot station. The cold station can cool to ice or down to 16. The hot station, which is here, can heat to 30, 37, or 42 degrees Celsius. And then on the side here, we've got an incubator, and that's where we're going to be putting our Petri dishes. And so in this case, I'm going to press the 37, all right? Oops, I'm going to press the 37 so that it turns on, and now the incubator is going to start heating up, okay? It can take about an hour for this incubator to warm up because it goes slowly because you're most likely adding kind of cool plates to it. All right, so I'm going to open the door here. Now within here we have what we call humidity chambers. And so what you can do is you stack your Petri dishes so that the lids are down, right? So bottoms are up, lids are down, and that helps to prevent evaporation of liquid within your agar. So you put them upside down on their lids. Then you can put the humidity chamber on, and then you can simply slide them in like a little pizza oven here, okay? And each of these playgrounds comes with 4 humidity chambers because you can fit 4 stacks or 8 plates. If you don't need all of them, just remove them, okay, and then only use what you need, and you can put all the other humidity chambers in a little container somewhere else. Okay, so that's really it. I'm going to close that. I'm going to lock it, and I've started my timer already. We're going to let these incubate for up to 24 hours, and it's really important that you don't wait any longer than that because once you hit that point, the cells just don't take in DNA as well. All right, the ideal time is is around 12 hours. Once you see these small colonies growing, 16 hours is still great, and 24 hours is the latest, all right? Some tricks that some teachers will use if you're on a very strict schedule for classes, for example, is if you start with first setting your cells at 30, so let's say you have a class in the morning, and then you've got another class like right at 24 hours later, or even a little more. You can grow your cells initially at 30 for like the rest of the day, and then right before home from work, you can turn it up to 37. And that way, they'll grow a little slower at the beginning, and then when you up it to 37, they'll grow at their normal pace again. But you've kind of like shortened how much they're growing by lowering the temperature initially. Okay. Alrighty, so we'll leave it at that for now. You can put your Petri dishes in the fridge. The stencil you can hang on to for later use. You're done with the day one bag. All of the loop packaging can go into the regular garbage because it shouldn't have touched anything. And this little baggie can go in. The rest of this can go actually into the fridge. Okay. Now, one quick note, comparing this individual engineer kit to the group pack. So the group pack basically comes with some shared resources, which include the DNA, the cells, which we just used, and the inactivation bags. And then there are 8 separate packs that will have the loops and the agar and antibiotics and the Petri dishes and whatnot. So really these 8 different packs can be passed around, and then the different groups just share the cells and the DNA for the most part. Okay? Alrighty, that's all for today. See you tomorrow. Hello, we're now at day 2, and the main goal of day 2 is to do the actual transformation or genetic engineering step, where we're going to collect some of our blank cells that we streaked out yesterday, and through a little bit of temperature changes and mixing with a buffer, we'll add some DNA, and that'll get into the cells. And then by the end of today, we want to have these engineered cells being recovered so that they're starting to grow again and divide again, and so they're able to continue on with their new engineered traits. All right, so we left off yesterday putting our cells into the incubator on our negative control selective plate and our non-selective plate with the blank cells. And we finished off with a bottle yesterday. There was a little bit of agar left. I didn't mention, but you can hang on to this. This is sterile, and these bottles are quite nice, fairly expensive, so you can clean them out and use them for other lab activities, right? And you can see there's some agar on the bottom, and you can just give it a little bump, and you see it just popped off. And you can actually even put that into your discard container. So if you just kind of shake it, it'll flop in there. And now your bottle, you can just wash it out, a little bit of soap, rinse it out, and then ultimately rinse it out with distilled water, and you're pretty good to go to use it for other things. All right, so let's just revisit what we have here in our package. We've got a couple of blue loops, and we've got a couple more yellow loops. We have our Petri dishes from yesterday. So we have our plus selective plate and our selective E or experimental plate where we'll put our cells. And we have our day 2 bag. Okay, so we're going to be using the day 2 bag, of course, but we're not really going to be using our plates again today because we're just getting to the recovery step, and we'll actually be using these on the 3rd day of activities where we put our recovered cells onto these Petri dishes and incubate them so that they can grow. So I'm just going to put these back. We're not going to need these today. We're not going to need our streaking stencil. You can hang on to that, use it for other times, and we're also not going to need these. We're going to be using these loops tomorrow when we streak our cells onto these plates. So I'll just leave those in there. And yeah, let's have a look at our cells and see how they grew. Just going to open up the door here. This little pizza scoop here. There we go. Come on. Oh, there's another humidity chamber in here. Let's get that out of the way. And let's get our cells. Okay. So here's our cells. So that looks pretty good. You'll remember that the first line had lots of cells on it. The second line you can see it dragged through, dragging less cells. And then the third line, especially the one that went across here, we're getting individual colonies. And that's exactly what we want. We want these individual colonies of cells that are growing quickly, and those are going to take up DNA the best, okay? And just to remind you about the time schedule, you definitely want to do this step before 24 hours later. Ideally, like 16 hours is great. And there was that hack that you can do if your class is 24 hours later or slightly more. You can first start by incubating at 30 degrees Celsius for the first, 6-8 hours, something like that, so they grow more slowly, and then you can increase the temperature to 37 so that they pick up and they grow rapidly, because it's the rapidly growing cells that take up DNA the best. Okay. You also remember our negative control plate, where we streaked blank cells on a Petri dish that has antibiotics. And the negative result is that nothing would grow because the blank cells don't have any resistance and the antibiotics would kill them. So as we can see, there's no cells growing despite putting lots on there. And this really signifies that the antibiotics are in there, they're active, and they're doing what they should do. So that's a great result right there. Okay. In tomorrow's exercise, we're going to have a positive control plate where we use the positive control cells within day 2 bag to streak, and in that case we would be expecting a result. We would be expecting these cells to grow because they already are engineered, okay? And so that would be kind of the opposite kind of control, okay? So yeah, I guess let's get to it here. So just to let you know, we're sitting at about 21 hours after the activity yesterday, okay? All right, so for today's activity, we're going to be using, we're not going to be using the plus cells, that's for tomorrow, but we are going to be using some DNA, a tube here called transformation buffer or T buffer, and this recovery media, all right? And whenever you're starting this experiment, you want to make sure all the liquid or most of the liquid is in the bottom of the tube. And you can either do that with your centrifuge by putting the tubes across from one another and doing a really quick centrifuge so that it's all at the bottom. Or you can do a quick snap, so you just kind of like do the whip it like this, and then the liquid will get pulled to the bottom. You can even like tap on the bottom. That'll cause some of the droplets to go down. Okay. Go, some liquid at the bottom there, recovery as well. Okay, so this step is pretty straightforward. Just follow the instruction manuals, but we're going to need the cold station, so I'm just going to turn on the cold station here so that you can see that there's ice and there's 16 degrees. You definitely want the ice, and you'll see the blue light come on. We're also going to be doing a heat shock, and so right now the incubator is on, but we're going to turn on the hot station here because we're going to need it at 42. If you turn that on right now, it will slowly cool down from its point. Give it 5 to 10 minutes. It should cool down to 42 Celsius, which you'll use for this activity. The cold station will cool down over a couple of minutes to 4 degrees. If the hot station is above 42 when you're doing the heat shock by like a degree or two, it's not a big deal. You can just go for it. All right, so you need to put your transformation buffer on the cold station, okay? So you're gonna put it in there, and this is to cool the whole reaction down. There's a lot of salt in there, especially calcium chloride, which can be harmful to cells. And so you kind of want to cool it down to help the cells survive a little bit better, all right? And so that'll help preserve the cells. I'm going to just put my recovery media tube on the hot station so that it warms up for the recovery step. You don't have to do this, I generally do. And we'll just leave the DNA here. Okay, so once it gets down to 4 or so, between 3 and 4, just wait maybe 20 to 30 seconds before you move on to the next step, and that'll allow the tube to cool down and the liquid to cool down inside of it, all right? I'm going to remove the lid here. It's pretty much the amount of time it takes to get your other stuff ready. So I'm not going to like just pause here. I'm just going to move over and move on to the next step. All right, so we're going to be collecting some colonies because, again, they grow fast and they take up DNA the best. So you want to avoid the lawn areas here. A lawn is where you have like this solid growth of cells, all right? So we're going to avoid that. So we're going to try and collect maybe some of these small colonies right here, maybe some of these up here. But really what you need to do is collect enough cells so that the center of your little blue loop end here is full of cells. So you don't need that much cells. And when you look at this, actually maybe like 1 or 2 colonies would be fine. It really depends on how long you've let this grow, right? So if I looked at this after 12 hours, these colonies are going to be much smaller, maybe even half the size. And so I might need to collect more colonies in that case. But the goal is to have like a nice little bunch of cells filling the center of this loop, okay? So again, just pop the back out there. And let's just have a look here. So yeah, I'm just going to go for these small ones here. Might be a little bit wet, so it might just drag on the agar. Go this way here. It's wet, so you can see that now the center is kind of full of cells, almost. I'm just going to collect one more colony here. Alright, so that's pretty much full, so that should be good. And now I'm going to go back and I'm just going to, just like the game operation, I'm going to put this in so that it gets to the bottom of the tube. And then I'm going to spin the loop here, and that's going to dislodge the cells from the loop, and it's going to mix them in. The goal here is to have all of your cells suspended or nicely mixed in, and your solution should be cloudy. So if we look at it, it's definitely cloudy now, right? All right, so we don't want to wait too long. We want to do the next step as quickly as possible. So maybe mix for 10 seconds and move on to the next step. I just put my loop in the discard container. All right, the next step is getting DNA. This is really straightforward. You're just going to dip your loop into the liquid at the bottom. Just spin it around so that the center of the loop fills with the DNA that's inside that liquid. If you have a look right in the little loop end, you can see the light distort a little bit. That means the liquid's in the middle. I'm just going to go back and I'm going to put it in and mix it for about 10 seconds. And I'm going to reset my timer here because now I'm going to let that sit for about 5 minutes. It's a little flexible here. I would say between 5 and 10 minutes. But 5 minutes is good. There we go. Put that in the discard container. I'm just going to set the lid back on and let this stay cool. You can see the hot station is almost down to 42 now, so it should be in a good spot for when we do the heat shock, which is the next step. And what happens in the heat shock step is right now it's cold, and you can think of the membrane of cells to be something like if you ever cook with oils, especially animal-based oils on a stove, like if you make bacon or sausage or something. And after you're done cooking them and you let the pan cool, it congeals and it creates this like thick, I guess it's also similar to like butter or margarine, right? At room temperature, it's a solid, but if you warm it, it'll melt, right? And so right now at 4 degrees, the membrane of the cells is a lipid, and so it's going to be quite rigid. It's going to be very intact, and the DNA right now is kind of floating around outside of all the cells for the most part. It's mixing with some of the calcium, and the calcium helps to cause the DNA to get smaller, to kind of bind to itself, but also bind to the surface of the cells. But again, the membranes are fairly rigid because of the cool temperature. So in the next step, we're going to move these cells to the hot station for 90 seconds, and this is going to warm up the liquid with the cells, and the cells are going to warm up. The lipids in the membrane are going to move around a lot more, become more, we'll say, liquid in a way, and that's going to make the membranes more permeable, meaning that things can get in and out of them easier, including the DNA, and so that'll help the DNA get into the cells, all right? And then after those 90 seconds, we're going to move it back to the cold station. so that it cools down the membranes, traps some DNA inside, and then the last step is we're going to pour some of this recovery media into the cooled down cells. This is going to dilute the calcium so that it's not harmful to the cells, and it's also going to provide the nutrients, the amino acids, the sugars, the minerals that these poor bacteria need to grow and survive, all right? And then that'll be the last step. We're going to let this go for up to 24 hours, okay? And then day 3 will be taking these cells and putting them onto your Petri dish, okay? So I'm just going to clean up my space here. You can hang on to the DNA. You can put it into a fridge. After you've used it, it should last like 6 months if you leave it in a fridge, but you have like opened it to the air, and so things could have gotten in there which might cause it to break down a little bit more, but it should stay stable, and so you can collect these and get more and more colors. The cells we are done with. That was all. We only needed a few colonies of these bacteria in order to do this experiment. So I'm going to go ahead and put that into the discard container. Okay, you don't need to remove the lid because bleach can get in there. The selective plate you can also put in there, the negative control, that's finished. Oh, you can also take pictures of your Petri dishes, right? You can log them in your logbook, or take pictures every step of the way so that you can log your journey and keep it sort of as a journal as well. So take pictures, but yeah, ultimately this plate can go into the bin as well. Just like yesterday, these sterile loop packaging, they can just go into the garbage. They don't need to be put into the inactivation bag because they are sterile. So these can go into the regular garbage. Okay, I'm just going to go ahead and put the DNA back into the day 2 bag where it came from. And I'm going to put that back into this bag, which will go back into the fridge for tonight. Move those there. Okay, so we're at 4 minutes and 37 seconds. Temperatures are pretty good. Alright, we're nearing 5 minutes. Alright, so 90 seconds. I just move it to the hot station here. And so again, right now the cells are warming up. The membranes, which are made primarily of lipids, are kind of vibrating more. They're becoming more quote-unquote liquid, and so that again makes the membrane more fluid and permeable to things in the outside environment. So right now maybe the calcium is coming in due to diffusion. You might have learned about diffusion, from high concentration to low in the cell, which might be pulling the plasmids into the cells. Funny enough, the exact mechanism behind this transformation isn't very well understood. So there are a number of hypotheses that are still out there. All right. We're at 6 minutes. $6.21, $6.22, okay, we got to get ready to shift back here. 25, 26, 27, okay. All right, we shift back onto the cold station. Now the cells are cooling down. Again, the lipids are cooling down, making the membrane more rigid. The DNA is trapped inside the cells. And so we're just going to let this cool for 2 minutes. So at, that was 6:30, at 8:30, we're going to move on to the next step, which is to pour this recovery media into this tube, and then we're going to mix it and incubate the tube on the hot station at 37 degrees for up to 24 hours. You can do it overnight or up to 24 hours. Okay, so again, this is going to slowly cool down to 37. If it's still above 37 when you reach the next step, that's no problem. It'll cool down pretty quickly, and it's not going to harm the cells in any way. Again, you want to try and make sure all the liquid is at the bottom of the tube, not in the lid, so that you get everything in there, or as much as you can anyway. Alright, so the cells and everything should be nice and cool in there. DNA should be trapped inside of the cells right now. I'm just going to get the tubes ready. I'm going to just take the lid off of our cells. I'm going to take the lid off of the recovery. We've reached the 8 minutes and 30 seconds. And I'm just going to put it over top like this and on the table, not on the playground. I'm just going to give a little tap on the table and we're going to get pretty much all of it out. That's, you got plenty out if you see this little amount remaining. Okay, so you won't be able to get it out, all out, and we've accounted for that in the volume. I'm going to put the lid back on and you're just going to mix it. You can give it a little shake like so. There we go. Simple as that. Now your cells are mixed into the recovery media, which again has diluted the calcium so that it's not such a high concentration and dangerous to the cells. And we've added the growth media, right, which is the sugars and minerals and amino acids and things like that. So I'm just going to give it a little whip, make sure all the liquid's in the bottom, and we're now going to set this in the hot station for, again, up to 24 hours. You can see it's still at 41, but that's no problem. It's going to cool down pretty soon. I'm going to turn off the cold station. All right, we don't need that anymore. And I'm going to go ahead and reset my timer so that it's now from the point where we finished the recovery step. And yeah, that's pretty much it. I'm going to put the recovery media tube in the inactivation bag. I'm going to close my inactivation bag because we now have some Petri dishes in there and we don't want to promote any sort of possible growth of contaminants floating around in the air, right? So if you put your Petri dishes in there and you have some mold or something flying around in the environment, they could still land in those Petri dishes and grow, and you just don't want any weird growth happening. So if we close the bag, that'll help limit that. We don't need the incubator at all today, so I'm just going to leave these here, and we're done with this for today. We can put this in the fridge for use tomorrow. So that's all for today. So again, tomorrow we're going to be taking our recovered cells, we're going to be putting them onto these Petri dishes, or one of the Petri dishes, along with a positive control, and then that's called plating, and then we're going to incubate them, and on day 4 we should have some engineered cells where you've added a DNA plasmid into the cells, and that DNA plasmid is expressing genes, notably that are going to create some colorful pigments. Okay? Alrighty, see you tomorrow. Hello, it is day 3, and today we're going to take some of our recovered cells and put them onto the Petri dishes we made on day 1. So just as a quick recap, day 1 we made some Petri dishes. We made some selective and non-selective Petri dishes. The non-selective are the plates that don't have any antibiotics. The selective do have antibiotics. And then also on the first day we streaked some blank cells, meaning cells that have not been engineered yet. We streaked them onto the non-selective plate so they could grow. We then put those into the incubator. And then we also did another plate, the negative control plate, which does have antibiotics in it. And we also streaked some blank cells onto that and incubated that as well. And then on day 2, when we came, we saw that there were some nice colonies of blank cells on the non-selective plate. When we looked at the negative control plate, we saw no growth of bacteria at all. And this is expected. This negative result is expected and suggests that our plates were made properly, or at least they have antibiotics in them, because the blank cells don't have any gene in them. They haven't been engineered yet with a gene, so they become resistant to that antibiotic. And so this is all good. And then on day 2, what we did was we actually engineered some cells. So we collected some cells, a couple of colonies from the non-selective plate, and we mixed them into a transformation buffer that was on cold. And then we added some DNA plasmids to that, mixed them, left them on cold for a short while so that the plasmids could start interacting with the cells, and then we put it onto the 42 degrees hot station during what we call a heat shock, which warms up the cells, causes the membranes to become more permeable, the DNA can enter the cells, and then we put them back on cold to cool down the membranes, make the membranes more rigid, trap the DNA inside, so that your plasmids stay inside, and hopefully the cells will continue to read them and create the colorful pigments and the antibiotic resistance. And then at the very end of yesterday, or day 2, we mixed those transformed or heat shock cells with some recovery media in order to dilute the high concentration of salts, namely calcium, so that the cells could withstand the calcium, but then also to give those cells some nutrients, sugars, amino acids, etc., so they can start growing and dividing again. And then we put those at 37 degrees Celsius overnight, and here we are today. So we have our cells. Some of the way through, it's always good to grab your cells, give them a shake. That will just make sure they're mixed and suspended and still kind of floating around, able to eat those nutrients and continue to grow and divide. All right, so now we're going to do the last step, the day 3 steps, which is essentially to pour half of our engineered cells onto the Petri dish and spread them out. You only need half because that should be plenty of cells in there. And then the other thing we're going to do is we're also going to streak or plate out some positive cells onto the plus plate. And the positive cells are already engineered cells, all right? So they already have the antibiotic resistance. They already have a plasmid that can create a particular color pigment. And the positive control is similar to the negative control, except it's sort of the opposite in that in this case we would expect a positive result, which would be the growth of the cells, right? And so the reason for having a plus plate here is, what if there were what if there's too much antibiotics, for example, right? You put your cells on here, they don't grow because there's just too much antibiotics, and they just kill all the cells because the cells don't have enough resistance, right? And so that's one example of why you'd want to do a positive control. Okay, because if your cells don't grow tomorrow, you kind of want to be able to troubleshoot that, right? And if your positive control also had no growth, then a really good indicator might be that there's too much antibiotics. Okay, so that's what controls are for. They're there to help you understand the boundary conditions of your experiment and troubleshoot if need be. Okay, so you may have noticed there's a little bit of liquid in one of these plates. It's just from condensation, because when we made these plates we put the lids on and then we put them in a fridge. So one thing you can do is you can dry your plates a little bit. That'll definitely help you in this next step where we want to put our cells on and then have the plate dry a little bit. Now, one trick to be able to do this kind of rapidly is to actually use the fan of the cold station here. And so you don't want to pour liquid in there. In fact, you don't really want to put liquids in open containers on top of the playground in any case. But what you can do is, I'm just going to turn it on to 16. You're going to feel some air coming through, and it's going to end up being warm air. And just put your lids over top like this. The air is going to kind of help heat up the plates a little bit, and then also cause some evaporation. Another way you can do it is you can actually put them inside the incubator of the playground for a short while, and that'll also warm them up and help to get this next step going. Okay, so I'm going to leave them there for a few minutes. I'll tell you a little bit more about the positive cells here. So the experiment that you are doing now, and especially that you did yesterday, namely the transformation of cells with a plasmid, that plasmid is a small circle of DNA. Okay, so you can imagine there's like these tiny little Fruit Loops of DNA that were in this tube here. Okay, so this is the tube that I used yesterday, and so there's a little bit of liquid down there, and you might recall we only took 1 microliter of it. And so you can't see the plasmids. They're really small. They're at the nanometer scale, and yet those little tiny loops of DNA, they typically have 3 important factors in them. One would be the gene of interest. So a gene is a snippet of DNA or a segment of DNA that is ultimately responsible for creating something in the cell. All right, and this could be a colorful protein, like what we're doing in this experiment, or it could simply be some RNA, right? So, but really a gene is a unit of DNA that has all the information that the cell needs in order to create something, okay? So there's the gene of interest, our color-producing protein. There's also another gene, which is called a selection marker, all right? And there's many different kinds of selection markers. There's lots of different kinds of plasmids, but often these selection markers are a segment of DNA that will result in the creation of a protein enzyme, okay? And so that's a protein that's able to cause a chemical reaction. And that protein enzyme is typically able to break down an antibiotic, okay? And so because this plasmid also has that gene in there, it will allow the cells to be resistant to the antibiotics in the plate. It'll cause the creation of a bunch of protein enzymes, which are kind of like a defense layer within these bacteria cell that protect them from the antibiotics. They keep chopping down the antibiotics as they are entering the cells, okay? And then the third piece is called the origin of replication. And you might have learned about this. This is the point where the cell will recognize that the DNA plasmid and actually copy it to create 2 DNA plasmids, all right? And it's really neat because you can have lots of different origin of replications that will tell the cell to keep different numbers of these plasmids within the cell. So for example, keep 2 plasmids in the cell, or keep a few hundred plasmids in the cell at any given time. And this is really important because you have to remember that the cells are going to grow and divide, right? And so if that plasmid you add also doesn't divide, eventually the cells that split off, or as they split through binary fission, one of the 2 cells wouldn't continue to have any plasmids, right? So if just your 1 plasmid goes in, and then that 1 cell divides into 2, only one of those 2 cells is going to get your plasmid, right? And then during the next division event, same thing happens. With the 1 cell, only one of the 2 will get it, but then the other cell that didn't get it before is just going to split into 2 cells which doesn't have it, right? So in the end, you're only going to have 1 cell that has your plasmid, okay? But because within this plasmid you have an origin of replication, you have many copies, so every cell that divides, the daughter cells get a plasmid, and they can continue to replicate and copy those plasmids so that you can ultimately see your trait in thousands, millions, if not billions of cells, okay? All right, so these cells here, the positive control cells, they've already gone through this procedure that you went through yesterday and you're going through today. They already have a plasmid in them for creating the antibiotic selection resistance, as well as a color-producing pigment, a protein pigment, and then, of course, the origin of replication. Okay? And so when you put these on a Petri dish, the plus plate, that's sort of like your expected result, right? Because these are already engineered. You're just going through the engineering step now, but ultimately the cells should be similar. They should be colored. And while the colors that you got, the DNA plasmids you got, they may not produce the exact same color as the positive control. It's more or less you need to consider how the cells grow, what they look like, and do they create a color. Okay, so let's just set that down here. We're going to need 2 loops. Again, 1 for streaking your engineered cells on the Petri dishes, and then 1 for doing the positive control. All right, it looks like the condensation is now off of the lids, so that's good enough. Let's move on with our experiment here. Maybe I'll leave this the experiment one on there momentarily while I do the plus. Okay, so we're going to do the plus first, and it's as simple as dipping your loop into this tube like you had done with the blank cells, and then except we're just going to rub it all over the plate. Okay, so it's very much just like when you did your negative control. You can see, you might be able to see some colorful cells in there. So again, you just need to touch the top. You can see that this is wet. Okay, so that suggests there's lots of cells on there. And I'm just going to put this on here. You can draw a picture if you want. It's a little difficult when it's this wet to draw a picture, but I'm just going to spread them out a little bit like that. It's fine. Whatever shape you want. There's no special streaking method here. Again, the idea is just to put cells on a plate and see if they grow. All right, now I'm going to put my loop into the inactivation bag. If you're in a classroom, you might just have a yogurt container and you can just put all your stuff in the container or your discard container, and then at the end of the class you can pour your waste into the inactivation bags that were in your kits. Okay, so this one's done. That means my positive control cells are also done. I'm going to put those inside of the inactivation bag. And now we're on to the last part. This is the grand finale. This is what you've worked hard to get to. So again, we want about half of the cells within the tube. If you are using pipettes, you can pipette 150 microliters is a good amount to put on the Petri dish. Even 100 is fine, but roughly half. So we're just going to give it a little... There we go. Just a drop like that is good. That might be a little bit less than half even. I'll give a little bit more here. Whoop, there we go. Well, that's more than half now, but so be it. So we're done with these cells here. So yeah, you can see I probably put in about 2/3. It's okay though. It just means it's going to take a little longer to dry. Okay, now I'm going to take my loop, and you always want to be using inoculation loops at a kind of a sharp angle, right? So you kind of want to be down, working kind of at this. If you work pointed kind of downwards, or if you try and spread like this, you're going to poke into the agar really easily. So I even kind of like touch the edge of the Petri dish and kind of just rub around, and you can spin the plate with the other hand. And eventually it should cover everything nicely. There we go. The plate is covered. And you can see that there's definitely liquid in there, right? And so that's what we need to allow to evaporate now. Okay. So I'm just going to put my loop now into the inactivation bag. I'm going to put this back on the cold station here like so, and let the air blow over it and evaporate that liquid, and then the cells are going to be adhered to the surface of the agar. Okay. So this process can take 15 minutes or so. It really depends on how hot your environment is and how dry it is. All right, so just kind of got to be patient, because ultimately we need to flip the Petri dishes over to kind of lock in humidity, and so we can't flip them over right away, because then your cells in that liquid are just going to pour into the lid, right? And then they're not going to be on the agar, and your experiment won't be so successful. All right, so I'm just going to turn off the camera now, and I'll be back maybe in 10 or so minutes once this dish is dried. You can, while you're waiting, you can kind of turn it a little bit every once in a while. Just kind of give it a little shift over. Again, you do not want to spill any liquid inside of the machine, or you are not going to be happy, or your teacher is not going to be happy. Okay, so I'll be right back. All right, so I'm back. It's been about 5 to 10 minutes. It's kind of dry in the environment I'm in right now, so it's evaporating pretty fast. Kind of, if you tip it around, you don't really see much cells moving around. Virtually no liquid. So now you're in a good place, if yours looks like this, to flip it upside down. Put the lid on and flip it upside down. I'm going to turn this off. Okay, so now you're going to be incubating your plates. So I'm going to go to the incubator tab here. I'm going to press start here. Come on. There we go. Okay, so incubator is on. So I've got my little paddle here. Flip your plates upside down, and again, that's because, so the top is underneath here. As this heats up, 37 degrees is a pretty hot environment. It's going to cause evaporation of the water that's in the agar, and by flipping upside down, it helps to trap the evaporated water in the Petri dish and keep the surface of the agar nice and humid so the cells grow. Okay, if you were to incubate them like this, slowly or much more rapidly, still slowly, but much more rapidly than if you flip them, the plate will actually dry out and it'll thin out, thin out, thin out, and then also the surface of the plate will be a lot drier and the cells won't do so well. Okay? All right, so you put on the little paddle. We're going to put the humidity chamber over. That's to help lock in some of the humidity. Unlock the playground. Just going to put that in, and you can fit up to 4 stacks, so 4 groups of students, or you can do 4 experiments at the same time if you want. Again, if you're not using 4, you can just put in however many you need, and so in this case I just pushed it into the front here. I've got a blue LED running inside right now, which doesn't need to be on. I think it just looks cool. So I'm just going to close the door, lock it, and I'm going to start the timer. So it's going. Now these cells should grow and divide, and you should start to see them in less than 24 hours. They'll be small colonies. You may start to see them changing colors around 24 hours, but definitely wait 48 hours to really see them grow into larger colonies and change color. The pace at which they grow is quite a bit slower than when you did the non-selective cells, because the non-selective cells, they didn't have any plasmids in them, so they weren't being asked to create something, right, like protein enzymes or colorful proteins, which can take some extra energy from the cell's normal metabolism. And then 2, the antibiotics, they do slow the growth of the cells as well. So because these cells are now growing on antibiotic-containing plates, they will just grow slower, period, because there's this battle going on between their defensive forces, namely the selection marker protein enzyme that you've engineered into them, and all of the antibiotics in the LB agar that you added on the first day. Okay, so wait 48 hours. You can look in between and see how they're doing. Take some pictures in between, maybe see how they grow through time as well. And usually up to 72 hours. 72 hours should be more than enough time to start to see them, okay? If you're growing these on a weekend, like let's say it's Friday now, and you're not going to be back till Monday, so that's 24, 48, 72 hours, you could leave it at 37, but you can also grow them at 30 degrees if it's for longer periods of time. So 48 hours is recommended because the plates still do dry out. If you're doing 72 hours or even more, I'd recommend that you can just have them grow at 30. They'll still continue to grow and divide, but importantly, the plates will not dry out at that temperature, because if the plates dry out, the cells are toast. Okay? All right, so we'll leave it at that for now. I'll check back in about 24 hours and maybe 48 hours and 72 hours, and we'll see how the results are going. Actually, on that note, though, the positive control cells, they should grow within 48 hours for sure, and you should definitely start seeing growth at 24 hours. And this is because, so another thing I didn't mention was when you engineer them, they've gone through this kind of horrible process where you heat shock them in salt and you're recovering them, and so they're also more slow to grow than cells that haven't just been engineered. And the positive control cells that you used, they were engineered long before they were put into that tube that you took them from, and so they're kind of in a good state for growing, right? They're kind of ready to go. So when you put them on the plate, they're happy to keep growing on that selective plate, whereas the cells you've just engineered, they're still kind of in rough shape from the engineering that they just went through. okay? And that's why those will take a little bit longer to grow. All right, so see you in 24 hours or so. Hello. So we are now at day 4 of the engineered kit experience, and essentially today is looking at some results. So just as a quick review, on the first day we made some Petri dishes, 1 non-selective, 3 selective. We then streaked some blank cells, meaning cells that haven't been engineered yet, onto the non-selective plate so that we could get some colonies. On day 2, we collected some of those colonies and we mixed them into a T buffer, a transformation buffer, which has calcium chloride, among other things, that help the DNA plasmid, to get into the cells. We then added a DNA plasmid and did a heat shock step, which was followed up by a recovery step, where we added some recovery media and let the cells incubate overnight to recover and start growing and dividing again. On day 3, we took some other selective Petri dishes that we made and we streaked or spread out our cells, our engineered cells, onto the experimental plate. All right, and then we incubated that about 20 hours ago. So we're about 20 hours after that step. And then all the while we did 2 controls. We did a negative control where we put blank cells onto a selective plate. And this was to test out whether the antibiotics actually work, right? Because the non-selective doesn't have antibiotics, and blank cells can grow on that, but can blank cells actually grow on the antibiotic plates? And we found that no, there was no growth, so that suggests the plates had antibiotics in them at the right level so that the cells couldn't grow. And then yesterday we also did a positive control where we put some pre-engineered cells, meaning cells that already have this DNA plasmid or a DNA plasmid with a selection marker in it, and so we spread those out on a plate, and we're going to see what those look like today as well. So I'm just going to open here, and again we're at about 20 hours and 29 minutes here. All right, so let's open that. Let's grab our plates here. All right, so I guess let's start by looking at the positive control, and we can see that engineered cells do in fact grow on the selective plates. So that tells us a couple of things. One, it tells us there's not too much antibiotics, because you'll remember that the plasmid has a gene in it that creates a protein enzyme that breaks down the antibiotics as they're kind of entering the cell. And so they're kind of like a defense, right? And there's only a certain amount of enzyme produced, and so you can actually overwhelm the cells if you have too much antibiotics, and then the cells just simply won't grow and survive, okay? So this suggests that the Petri dishes were made properly when we compared the negative control, where non-engineered cells didn't grow, and then the positive control where engineered cells do grow. It tells us that there's the right amount of antibiotics and selection, okay? Great, so that's fantastic. And then, here's our results plate. So this is a Petri dish. You can see there's quite a few colonies on there. So this is a good result. Getting a single colony of bacteria that are engineered is actually a great thing. So if you got 1 colony, that's a start. I've been doing this for years and years and years, and so that definitely helps with me getting this result. But if you start with 1 colony, then the next time you do it, you're going to get maybe 10 or more colonies and more beyond that. So if you got a single colony, congratulations on that. As I mentioned before, it takes typically 24 to 48 hours to really start to see the colors emerge. We can already see that there's some colors here, so that's exciting, but we're definitely going to keep this incubating for another 24 hours or so because these colors are only going to get more prominent, okay? And I'll talk a little bit more why we have different colors here tomorrow. I'll tell you more about the DNA plasmid or the DNA that we used on the second day, okay? So I'm just gonna put these upside down, put the chamber on. Let's put that. Lock the incubator. So now that we got what seemed to be some really good results, we don't really have any need for this tube because this still has some likely engineered cells in it, right? This was our tube that we recovered our heat shock cells in, and typically it's good to hang on to that until you see a result. So if there were no colonies on this Petri dish, on the Petri dish, you could always pour the remainder of this onto that and onto your selective plate and incubate it longer, because maybe there's still some engineered cells in here that just didn't make it onto your plate. Okay, but now that I got a lot of colonies, experiment looked like it went quite well. I'm just going to take the lid off and I'm going to put it into my inactivation bag. Okay, yeah, we're basically done here. We're going to let these incubate a little bit longer. Always remember, take pictures of your plate so that you can log your journey as you go. And yeah, we'll leave it there for another 24 hours. Bye. Hello, so it has been 51 hours and 13 minutes since the plates with the recovered cells were incubated. So I'm just going to open it up here. We looked at the cells at around roughly 24 hours. Let's see what this looks like now. There we go. Look at that. So you can see the positive control purple cells are way more purple than they were before. All right. And maybe I'll just make a quick note about this. So you might remember when I was streaking, I kind of streaked mostly in this area here, right? I definitely did not take my loop and streak around this edge down here. And so if you ever see that, that's generally because there was some water or liquid on the surface of the plate. And so then you went ahead and streaked, and a little bit of bacteria might have gotten some droplets. And then as it's handled, especially if maybe it was rolled around, that droplet, which has bacteria, rolled around here and deposited cells as it rolled around. So if you ever see that ring, that basically means there was some liquid on the surface. All right, and that's something you need to be particularly keen to find in bioart, because if you're making bioart and you have liquid on there and you draw something, you know, you'll get these unintentional lines of cells. Okay, so yeah, that's what positive cells look like, and positive cells could be any color. Really, the whole point is that an engineered cell that is producing that protein enzyme that breaks down the antibiotic is being expressed, and then the color trait is being expressed. Okay. All right, and so now we have our experimental sample, and so this is from the actual DNA that we transformed the cells with. And so you can see there's actually many different colors here. We've got some fuchsia, we've got some orangey yellow, we've got some blue, so a few different colors. So that's pretty exciting, and there's I haven't counted, but there's got to be between 15 and 100 colonies there, maybe a little bit more than 100. So that's a pretty good result. All right. And so you might ask, like, how do we get this result? Why are there different colors? And really the reason is because the DNA we used is a mixture of different plasmids. All right, and those different plasmids have the same selection, so they can all withstand this antibiotic, but they have different genes for the color pigments. Okay, so they're just expressing different colorful pigments, and when you transform, a typical cell will take in around one plasmid, maybe a couple, but if a couple are in the cell will generally grow and divide and lose one of those two plasmids. It's just a natural thing that happens, and you're basically left with bacteria with one of those plasmids, and the colony ends up representing, you know, growth from that single bacteria producing those colored pigments. So that's why we get different colors, is because we just had different plasmids in that tube. Okay, so that basically wraps it up. Maybe just a quick summary. On day one, you created some LB agar Petri dishes where you mixed some LB agar powder with some boiling water so that the boiling water could dissolve that agar. And then once you poured it into the plates, it cooled down and solidified into like a jello-like substance. You made a non-selective plate and 3 selective plates on that first day. And the non-selective plate was used to streak out some blank cells. Again, those are cells that aren't engineered, so that you could get single colonies of those bacteria. On the other selective plate, you did a negative control, which you streaked with blank cells. And in that case, the negative control was that the blank cells should not grow because of the antibiotics. And that's exactly what we saw after we incubated those for about 16 to 20 hours, if I remember correctly. And then we collected some colonies from that blank cell plate, and we mixed it into a transformation buffer on cold. Again, the cold to help protect the cells. And then we added some DNA and left those for about 5 minutes. And then we did a heat shock where we warmed up the cells so the DNA could start getting into the cells. And then we cooled it down so that we could trap the DNA in. And then we added recovery media so the cells could start growing and dividing again. And we incubated that overnight at 37. And then on day 3, we plated our engineered cells. So on one of the selective plates, we just poured about half of our cells and spread them out and then let the plate dry. And on the other, we spread out the positive cells. And you might recall you got a tube of positive cells. All right, and then we incubated those. We're now at 50 hours, and we get a lot of colonies. And just to emphasize, a colony is a bunch of bacteria, right? So you're talking thousands to millions of bacteria that all started from a single one, okay? So when you did your transformation, a single bacteria took in that plasmid, and when you spreaded that single bacteria out on this plate, it grew and divided and grew and divided. So that one cell grew into thousands, if not millions of cells, right? And that's what a colony is. It's a mound of cells that originated as a single bacteria. And so if you counted all these colonies, that would give you an idea of how many bacteria took up plasmids within your transformation mixture of cells and DNA and all that. Okay, so that's a fun exercise you can do, is simply count the colonies and see how many bacteria. But remember, you only poured about half of your cells on the plate, and so you don't have the full story if you just were to count these colonies. Alrighty, happy genetic engineering.